Good afternoon, good morning, everybody. I'm very excited to open up our webinar, the first of our 2019 webinar campaign, which is focused on improving productivity in cell-based, adherent cell-based bioproduction applications. So let's give it a start. As an introduction, we're going to scope the topic. I'm going to give you a few words about our division at Thermo Fisher Scientific and what we're doing, and walk you through the agenda for this presentation. So indeed, if you're interested in adherent cell-based biomanufacturing, the scope is that human vaccines, animal vaccine, and allogenic cell therapy, as well as vectors production, are the key areas where adherent cells are being used. Obviously, most bioproduction animal cells are naturally adherent. They're generally part of a living organism, and therefore their adaptation to suspension is a high level of technology, which is not trivial at all. So more often than not, it remains very important to be able to perform bioprocessing operations on cells which are still in their adherence state, because they have not been adapted to suspension or sometimes cannot be adapted to suspension. Right. Also, another framing comment to that talk is that the process economics are under increased pressure on a constant basis. Obviously, adherent cells are used to manufacture animal health vaccines, for instance, which is a form of bioproduction where manufacturing cost is of the highest importance, and also in areas such as human vaccines, where the quantitative needs related to pandemics and governments having to fund such to pay for such bioproductions is something very, very important, right? So today we're going to cover in this introduction, I'll provide you now in the 5 next minutes, a quick review on the two-dimensional systems for adherent cell culture, with a little bit of news in that space, but relatively mature technology. After that, we're going to have a more in-depth technical presentation by Alex Boscolo about the move to 3D cell culture for adherent cell systems. After that, you'll have a presentation related to the most recent advances from the cell culture media side with Céline Martin. And I will close with a short overview about novel downstream options which solve unique challenges in the area of adherent cell-based biomanufacturing. So in those first few minutes, let's have a quick review about a little bit more mature technology approaches, but some news about them. Obviously, adherent cells, when they start being cultured, it will nearly always be in flasks, and then several 2D systems can be used until there is an opportunity to move to 3D. There will be roller bottles as well as cell factory systems. Let's have a quick word reviewing this technology legacy. So obviously in the area of vaccine manufacturing, for instance, you are maybe familiar with the general steps of growing adherent cells by a system of multiple flasks, with the cells able to adhere to the substrate. And this is obviously proven relatively old technology, which comes with slightly more modern options. So one of the options is obviously roller bottles, which is as well a rather mature technology. I just have a little bit of news for you about this. Some recent studies about the various polymer resins available to achieve adherent cell culture in roller bottles. So if you're interested in that particular approach to adherent cell manufacturing, there's a new application note available related to a broader choice of plastic resins for your applications. Obviously, scaling that type of technology is a challenge. So here's an example of a large-scale installation. And of course, that has been the reason beyond those water bottles and those flasks to come up with the more advanced technology of cell factory systems, which is also a very proven approach. As you know, the thermoscientific Nunc systems uniquely are ultrasonically welded, which means that there is no use of glue and associated extractables and leachables. And there has been some level of innovation in that area related to what is known as the high-density cell factories, as well as the easy-fill cell factories. We will be coming up with more advanced versions in the area of connection into closed systems. So stay tuned with Thermo Fisher in that area in the near future. And there's also, for some applications, scaling up under that format, the opportunity to achieve some level of automation with various automation systems for very high volume cell factory-based manufacturing. Now, on that slide, you can see three automation elements which are available: the automated cell factory manipulator, the cell factory shaker, as well as the cell factory incubator. And so further down in this presentation, we're going to discuss how those adherent cell culture can be scaled up from the cell factory systems, as an example, into microcarrier applications, put into benchtop systems, into single-use bioreactor systems, and how some unique harvesting technologies can be utilized to make that a successful workflow. And with that, I am introducing Alex Boscolo, our field application specialist for single-use technologies, who will take you through the next section.
Hello, everyone. Thank you, Jeff, for the introduction. And yeah, we will spend the next 15 minutes talking about really our approach when it comes to scaling up adherent cell culture. Obviously, when it comes to adherent cell culture, we had a glimpse of it in the past 5 minutes with Jeff. But one of the main challenges with adherent cells is really the scaling up of the processes. As long as you are working at lab scale, cell factories, they work perfectly, and roll bottles are also excellent. But scaling up, it's really challenging for multiple reasons. So with that in mind, we've applied our knowledge of bioreactor solutions to the adherent cell culture space. To get there, I would like to start with a little bit of a history of the single-use bioreactor platform, so that just for really to give an idea of how we came to the point we are now, when we think that this would be a very good solution for scaling up adherent cell cultures. We started about 15 years ago, almost in 2006, with the high performa single-use bioreactor. There was a very basic one at the time, 50 liter, 250 liter. And over the time, of course, we had a lot of feedback from our customers, a lot of work in our R&D team. So we had a lot of evolutions over time with our single-use bioreactors. We scaled up with different sizes up to 2,000 liter, which was launched in 2010. But we also improved the hardware and the consumables of our single-use bioreactor to make sure that we were always on top of the requirements of our customers, and to make sure that our customers were able to do their process in the best possible way. One of the major introductions was in 2014, the drilled hole sparger. It was really a breakthrough for gas delivery into the bioreactor. We had several iterations in the past, but the drilled hole sparger was really what enables us two things: to move to a single sparger, which was enough for pretty much every application, and also to later on, 2016, 2017, it also enabled to scale to launch the 5:1 high performance hub. So with a better turn down ratio, 5:1, meaning that you are able to work at just 20% of the total volume of your bioreactor. And when with all these improvements in the bioreactors we've performed over the years, we are now in a position where our basic bioreactor is really good, pretty much for every application. But in some cases, and adherent cell cultures with microcarriers is one of those cases, we are able to do a little step further and make a few more improvements, a few more adaptations to our system, so that it's really perfectly adapted for the scope. When it comes to adherent cells with microcarriers, the main requirements of this kind of culture are high turn down ratio. So very often you want to start with a low volume and slowly fill up. You need to be able to see that low volume, and it's very important to keep on mixing while emptying the bioreactor, so that the system stays homogeneous. And we need to have high mixing power. Well, not really high, of course, because the cells growing on microcarriers are not the fastest growing cells. So gas transfer is not the most critical part, or mass transfer in general is not the most critical part. But definitely what we want to achieve is to have a microcarrier suspended and a very homogeneous system with as low as possible shear to the microcarriers, because they tend to be pretty shear sensitive. So really low tip speed and enough mixing power to keep everything suspended and everything homogeneous. And how we got there? We made quite a few steps. The most important step, probably at least for mixing, is we decided to step up the impeller size. So normally, with our single-use bioreactors, we use the very traditional impeller diameter of 1/3 of the vessel diameter. That standard has been used for bioreactors since the very beginning of bioreactors. It's very proven technology, but it's not optimal for microcarriers, because it does require a higher tip speed to keep the system homogeneous. And really what we want to be, when you look at the left picture, you want to be above the black line. You really want to be in a position where you have enough power input to keep everything suspended and homogeneous, but not damaging your microcarriers. So we achieved that by using a larger impeller. So for a 50-liter bioreactor, for example, we took the impeller of a 100-liter bioreactor, which is, of course, a bit wider in diameter. And we got there. We were able to have enough power to keep everything properly suspended, and we didn't see any shear or any noticeable damage to the microcarriers and the cells due to shear stress. On top of that, so that was really the main design change, but not the only one. We'll see that later when it comes to consumables. But when it comes to hardware, we do have a few. We did have in the past a few iterations of our hardware. So in this slide, we just show a little bit what the history of our bioreactor is. So we started with the legacy 2:1 turn down ratio. When we moved to 5:1, we've changed the motor block angle. We made it a steeper angle, so that the impeller was a bit further away from the walls of the bioreactor, and this helped us making sure that even with an upsized impeller, we would not be hitting the bottom or the sides of the vessel. Also, for the very same reason, for microcarrier application, we moved to a 4:1, what we call a 4:1 turn down ratio, which means that we've shortened a little bit the drive shaft, again, to make sure that there was enough space between the impeller and the bottom of the bioreactor. There needs to be a space not only to avoid the impeller hitting the bottom, but also to make sure that there isn't too much turbulence and too much shear on the bottom side, on the bottom part of the bioreactor, so not to damage your cells in that area. Also, in these slides we do see a few other options. For example, for perfusion, for what we don't have for microcarriers, is a special version of the drill door sparger. again, those are cells that don't grow too fast, so the griddle sparger is perfectly fine. It does achieve the optimum balance of oxygen delivery and CO2 stripping in the bioreactor without having to move or to go to a different solution. In this next slide, we do see a few other enhancements we made to our bioreactor consumables. Again, some of them we mentioned already, so the standard griddle sparger, an upsized impeller. What's very important, of course, is to have a single-use foam sensor to make sure that there isn't foaming in the bioreactor, or there isn't excessive foaming in the bioreactor, which could, of course, hinder the performance of the bioreactor and cell growth. And what's most important is also an extra line for decanting. So when we work with microcarriers, at least the work we've done in our R&D labs and the work we've done together with some of our customers, it's very important over time to have media exchanges. Depending on the process, can be 1 vessel volume per day, half vessel volume per day. That really depends on the process, but we need to have a way to let the microcarriers decant, remove all the supernatant, and put in fresh media. And the extra line for decanting, it's really important, because we were able to put that at the right side, at the exact size, at the exact position in the bioreactor, so that it would be just above the microcarriers once they're settled. So we were able to remove as much media as possible and put as much fresh media as possible during the process. And the last part, the 6th point mentioned in the slide, is the cross-flow sparger. That's really important when during the seeding phase, so when you're working at very low volume, because what we've noticed is that when you work at like 20% or slightly more than that of the full volume, there's a lot of air, there's a lot of overlay on top in your bioreactor, and not so much media. And what we see there is that there tends to be a gradient and a CO2 accumulation just on top of the liquid volume, which is hard to remove when you only have an overlay aeration from the top of the bioreactor. So the cross-flow sparger allows us to really clean up any buildup of carbon dioxide on top of the liquid, and keep everything properly aerated. We did, of course, a lot of tests in our R&D with our R&D team, and we worked. We really were able to demonstrate very similar cell growth and very similar cell densities at different scales: 50 liters, 250 liters, and 500 liters. Working with C-lagXL microcarriers, and what we noticed that the most important things were the adhesion phase. So making sure that your cells at the beginning of the process really adhere to the microcarriers, and we've achieved that with an intermittent decantation and resuspension phase. So for a few hours, we would just mix it for some time, then let cells and microcarriers decant, so that the cells had the time to adhere to the carriers, resuspend everything, and do a few of those cycles at the very beginning of the process, and then make sure that the cells were really in the right place on the microcarriers and were able to grow. And we have achieved pretty good and consistent cell growth at every scale. What was very important, of course, was so far we focused on the bioreactor only, but it's a whole process. So you cannot start with a 50-liter bioreactor. There needs to be a seed train before that. What we work with cell factories on our lab. It's pretty standard in the industry. So it's very important to make sure that at every step, every seeding step, you are able to recover all your cells and remove the microcarriers in a very effective way. And this is where the Harvestainer comes into place. So the Harvestainer is a system that we've developed to make sure that to really make sure that the cells and the microcarriers were optimally separated, and to have the highest possible recovery of cells at every step of the process. So in combination with the bioreactor, which allows the trypsinization step in the bioreactor itself, we were able to have the cells, the cells removed from the microcarrier, so trypsinization step in the bioreactor. Then everything was moved into the Harvestainer, and the Harvestainer, it's sort of it's a bag. It has a bag inside the bag, and the inner bag has a mesh which will stop the carriers and will let the cells and the media go through. So we were able to to separate everything in a very efficient way, and move the cells and the media to the next step of the process. The Harvestainer comes in different sizes, so it can be used in different stages of the process. And the size is the size of the inner bag. So when you look at the 50-liter one, it can be used for very large bioreactors. And the main advantage is that it's a very simple system to use, and it's also very efficient. It does have very little hold up, so recovery is pretty high compared to traditional methods. And it's a single-use system. Again, when you're working in a single-use environment, being able to do every step in a single-use way in a closed system, it's a great advantage. There's quite a few advantages. Again, there's a lot less manual handling. It's possible to make all the connections either in a sterile way, so we don't have to worry too much about operator safety as well, because everything is closed and disposable. High yield. Again, cross-contamination becomes a non-issue because that's one of the greatest advantages of single-use systems in general, and cleaning as well. So there's no cleaning when everything is single-use. And so with this, we were really able to prove that it's possible to scale up the adherent cell culture in bioreactors in a very efficient way. Again, there's a few factors to keep in mind. It's not a straightforward process the first time you do it. It needs, especially when you're moving from cell factories or roller bottles to a bioreactor, the process is different. There's a lot more control possibilities in a bioreactor compared to the traditional system. So it gives you a lot more options, but of course you need to know a little bit what you're doing, and that's why we're also here to help. But again, it's really important to have enough cells to start with, because that, of course, cell growth is a bit slow for this kind of cells, so you cannot start with a very low seed. It's important to have an oversized impeller. That's what we noticed, to keep everything suspended with low shear stress. And the trypsinization step inside the bioreactor, it was a real advantage. It makes everything much faster and much more efficient, thanks to the removal of microcarriers with the Harvestainer. And this was the end of my presentation. So I'm really happy to give the reins of this webinar to Céline for the next step.
Thank you, Alex. So, hi everybody. Thank you for attending our webinar. As Alex mentioned, my name is Céline Martin, and I'm an FS at Thermo Fisher Scientific, specialized in cell culture media for bioproduction. So on the next part of our webinar, I will be focusing my talk on how to leverage media supplementation for adherent cell culture. So first, let's focus on the parameters that can influence cell adhesion. So cell adhesion is a complex process in vivo already, but in vitro, we have to take into account these three main factors: the cells, obviously, the surface, and the medium. One cannot be chosen independently from the others. A cell type, or even different banks of the same cells, can show different patterns of CAM, or cell adhesion molecules, on their surface. For instance, different ratio of cadherines or integrins, etc. Cells might themselves be able to secrete their own extracellular matrix glycoprotein, like laminin or collagen. And finally, internally, each cell type has a different cytoskeleton organization and cytoskeleton dynamic, which leads to a more or less adherent or loose phenotype. Then, surfaces available can differ in terms of the plastic polymer itself, but also the surface treatments applied to the plastic, like corona discharge, UV, or plasma treatment. So these different types of polymer oxidation methods will generate different ratio of functional groups as shown in the table here. So this groups ratio will have a key impact on the unspecific interaction with the cell membrane or with the adhesion factor present in the supernatant. Surfaces can also be functionalized or pre-coated if these factors cannot be produced by the cells themselves or can be provided by the media. So finally, the media. It can have specific adhesion factors already present when supplemented with serum. For adherent cell culture, salt concentration should be are important, so notably for calcium or magnesium that can have an important effect. And for 2D adherent cell culture, a special attention should be put on buffer system. So, for example, the Hank salt mixture has been developed for 1% CO2 in ambient air, while the Debelco buffers have been developed for 10% CO2. So, depending on your process, an optimized buffer system can, for instance, reduce the need for medium exchange. So in this talk, I will focus on viral cell line and viral processes. For this application, the medium has a different purpose depending on the stage of the culture. Up to infection, the medium needs to provide all necessary components to get to the optimal confluence faster. But after infection, the virus will be controlling the cell internal machinery. So a leaner medium formulation can be used to increase specific titer and decrease cost. So what are the options for adherent cell culture in terms of media? Historically, base media such as DMM can be used supplemented with serum. Peptone and hydrolysates have also been used as serum replacements. In the meantime, complex serum-free medium have also been developed, and chemically defined protein-free media are available if the process can be adapted to suspension. Each of these four options can also be supplemented with individual components, for instance, growth factors like EGF, nutritional supplements like Glutamax, or process additives. While choosing one of these options, there are different points to consider. One is the broadness and the specificity of the solution. The more defined a medium is, the more specific to a cell subtype it will be. Meaning cells will need to be thoroughly adapted and rebanked to a more defined medium. While in comparison, most cell types will be able to grow in a base medium supplemented with FBS. The second important point to consider for process development will be robustness and qualification. It is important to be aware that controlling lot-to-lot variation and process reproducibility requires more effort with complex mixtures like serum or peptone. All of these options are provided by Thermo Fisher Scientific, and I will go over each one of them to show our recent innovation as well as our ever ongoing efforts to minimize your qualification investment using these supplements. So let's start with the use of sera. Gibco is a leading serum provider since 1962. But which sera can you choose from? The most common species used for blood collection are bovines, but others are also available, like sheep, goat, horse, or pork. For bovine, blood is generally collected from slaughterhouses and then transformed. Serum is hence a byproduct of the meat and dairy industries. Depending on the age of the animal, when the blood is collected, the composition of the serum will vary as well as its price and availability, which are also variable depending on the country of origin. For older animals used for donor bovine serum or adult bovine serum production, the growth factor concentration will be lower. However, price will be minimized, and for a robust cell line, it can be an option to consider. For serum, the key risk mitigation is to have a high level of traceability. This is why Thermo Fisher Scientific is part of the International Serum Industry Association and also applies an exclusive screening method against BVDV. This allows us to provide low-risk batches of serum for bioproduction usage. Serum can also be customized in terms of treatments to meet your requirements. We have also recently launched a new catalog offering of US calc serum supplemented with iron. Depending on the cells, it can show increased performance compared to calc serum alone, and it is a more cost-effective solution compared to FBS. To start our poll, you will have a first poll question on what is your main challenges regarding the use of serum at the moment, and all answers from the audience will be shared at the end of my presentation. So after serum, let's go to the next level, peptones. Advanced bioprocessing is now part of Thermo Fisher Scientific. At Advanced Bioprocessing, we have a long history of developing peptones for microbial application dating back to 1895. Advanced Bioprocessing has also been a pioneer in developing animal-free peptone in the 90s to replace serum in mammalian cell culture with the Gibco and the BBL brands. With this experience, we now support 150 commercial products in the biopharmaceutical industry. But going back to basics, what is the definition of a peptone? The source material to create a peptone can be animal, for instance, milk or beef extract. It can also be plant, like soybean, or derived from microbial, for instance, with yeast extract. This raw material will be digested by chemical or enzymatic treatment to provide broken down nutrients to the cells. As summarized on the right hand of your screen, peptones provide a complex mix of components, which is also why they can help improve process robustness. They can protect cells from stresses, byproduct toxicity, or delay apoptosis. Given the broad range of peptone source and type available, peptones should be screened to find the right one for your application. In the area of risk mitigation, we have one of the most stringent and rigorous vendor qualification programs in the industry. But raw materials are not the only parameter. We also validate our peptone manufacturing process to achieve high lot-to-lot consistency by using mass rate profile to control the degree of digestion, but also other analytical measures to control carbohydrates, vitamins, and nucleosides. To ensure consistency, we implement in-process control for each step highlighted on this slide. As example, for hydrolysis, a specific attention is put on temperature and pH control, as these are the critical parameters for the digestion process. For filtration, we control tightly our target % solid, and we require each batch to achieve this target upon the first filtration. Otherwise, the whole batch is discarded. Apart from being very stringent on the manufacturing process, our R&D is also developing new ways to implement peptones to facilitate serum removal. Recently, they have been working on supplemented peptones for viral cells. Here you can see that using this supplemented peptone instead of 10% FBS, doubling time of viral cells can be decreased. Using a 2D plastic surface and EM base medium, results yielded very similar adherent phenotype with a low level of FBS, or on the right side, a higher confluence with tightly packed cells without FBS at all. So now we will go to our second poll question. What are you considering as alternate to serum in your culture? And for the final part of my talk, I will focus on Gibco cell culture media and supplements. At Gibco, we provide basal media, complex serum-free media, but also animal origin and animal origin-free proteins to both research laboratory and bioproduction manufacturing facility worldwide. Our two main catalog formulations for kidney-derived adherent cells are VP-SFM and Optimise SFM. They can be used with no or minimal adaptation for cells such as Vero, BHK21, MDCK, etc. We offer regions coming from either animal or recombinant sources, for instance, insulin, or proteolytic enzymes such as trypsin or EEE-select, but also improved formulation of basal media like advanced DMM or Optimem, because in terms of innovation for viral cell culture, we at Gibco are constantly trying to reduce the use of serum for the future of bioprocesses. As example, no serum-free media is currently available on the market for human diploid cell line, to our knowledge. So one of our more recent prototypes is called Diploid SFM and has been developed to culture cells such as MRC5, SV1, and CEF. We developed a two-part approach using the same base media and two supplements, one from animal origin, the other one animal origin free. The goal was to be able to sow cells banked in serum directly in the growth medium to minimize adaptation time. Then the production medium is AOF to help our customers adhere to WHO guidelines in terms of BSA per vaccine dose. As you see on the left-hand side, viable cell density are reduced in this growth medium. However, if necessary, that can be mitigated with low addition of serum at 1% or conditioned media. And more importantly, when cells were infected with VSV as model virus, the titers obtained were in the same log range or higher with only 1% FBS addition. To conclude this short overview, I hope you have seen that there are many different ways to upgrade and develop the formulation for adherent cell cultures. Two key points are: one, to tune your media formulation to your surface and your process, possibly by adapting your cell line. And two, to take into account media scale-up and risk mitigation for bioproduction. For both, you can use our non-GMP prototyping service called Gibco Media Express. It allows you to order and test modified formulation of a medium, and that can help secure a smooth transition from your R&D to large-scale manufacturing. We are a team of field application specialists and will be happy to provide guidance on to which parameters can be modified to improve performance to your specific process. And then we will launch the last poll question on media, just to focus on why adherent cell culture is your process type of choice, and you will see the results on your screen as well. I thank you for your time, and I will send the presentation to Geoffrey for conclusion.
Thanks very much, Celine. Very, very insightful. Also, I want to take the opportunity to let the audience know that you should find in the webinar a resource section in which you can have access to some literature. And don't forget, you have the opportunity to start asking your questions, right? So I see a first question from Nila Branjan. So we'll try to save a couple of minutes in the end to go through some of your questions, and thanks very much for that. Right now, as Alex highlighted, in that bioproduction adventure of manufacturing from adherent cells, Thermo Scientific comes to you with a complete solution, one-stop shop offering, and it goes, of course, beyond cell culture, beyond harvesting. It goes into purification and the key analytics that help you keep that process under control. So in the coming minutes before the Q&A, we're going to have a brief overview about those technologies and how they can apply to such types of applications. So really, in that purification and analytics space, it's clear that it comes with unique purification challenges, right? So many suppliers' portfolios for purification were not necessarily designed for virus purification, more like legacy mainstream applications such as MABs. And it can be a challenge for the community of virus manufacturers, viral vector manufacturers, human vaccine manufacturers, animal vaccine manufacturers, to identify suitable technologies which meet their specific needs. And in particular, in an area which is not always associated to adherent cells, which is the AAV gene therapy vector space, POROS CaptureSelect technologies really have already allowed to revolutionize that purification. So I'm going to walk you through some of those technical aspects related to the purification of whatever you are making from your adherent cells. So we could take the example of a VLP, for instance, of a virus-like particle, which would be a popular product to be made out of adherent cells. So whatever your product is, if it is for human injection, obviously you will go through a number of purification steps so that all the required impurities are removed. And by that, some of you are probably familiar with the process-related impurities, such as the residual DNA or the host cell proteins, as well as the product-related impurities, which can be variants of your product. And in viral applications, obviously, you do not have common empirical affinity solutions such as protein A. Therefore, more often than not, there's a need for a very high number of purification steps, particularly if the product is for human injection. It can happen, of course. Some of you may be working in the area of animal health, where usually the number of steps is much lower, and then you may be interested in identifying the single purification step that will make your process more effective, provided that you would only have envisaged one if you have resort to chromatography purification. So in the first case, where we're here on that slide, or a human situation, obviously rather than have to use several ion exchange or several heat steps, one after the other, one of the smarter ways can be to introduce affinity early. Okay, so affinity early puts you back in the situation where your colleagues in monoclonal antibodies are, with just protein A and limited polishing. And this is what our CaptureSelect technology allows, to have that kind of process simplification on any biomolecule, because it provides custom affinity. I'm going to give you a quick word about this. Other than that, for other steps, it can be polishing or it can be alternate modes of capture. Other solutions than custom affinity are also possible. So CaptureSelect ligands, they are really the tunable affinity solution, now very proven
in some segments of the market. It's been around since 2006, and it's currently in a commercial process, and a very large number of clinical processes. And it consists of using single domain antibodies as affinity ligands. It allows, in other words, all the benefits of immunoaffinity without all the limitations and issues associated to it. So those VHH single domain antibody ligands will then be attuned for desirable purification characteristics, such as mild dilution in some cases and others, and immobilized onto chromatography beads in order to make complete purification technologies, which aim at a variety of types of targets. And viral vectors and vaccines are some of the targets, which is what makes them such a useful tool for adherent cell-based biomanufacturing. So that is here actually a customer case study of a situation where there was a gene therapy vector which was being manufactured. The purification took, as you can see, all the steps that you can see on the top of the slide. It could be simplified with CaptureSelect technology to the scheme that you have at the bottom of the slide. And it's actually a statement from the users with whom we had been developing that affinity solution, that the process yield improvement was increased from 20 to 60%, and cost reduction was as high as a factor of 6. So an extremely powerful technology. And how do we make it happen? There's a number of steps, right? So single domain antibody, they are naturally a part of the camelid immune response. So in some cases, and less cases, there's a need to actually immunize, for example, a llama with the target VLP or virus, and generate ligand libraries. But since 2006 that this technology has been used, there's a broad array of libraries, and more we can focus on the subsequent steps. And as you can see from the timelines at the top of the chart, it's not a particularly long process to end up with a fully documented bioprocessing resin that actually allows to purify your particular biologic. And this is a slide about the latest biologics on which we've been working, and as you can see on the right hand side in the viruses and the vaccine area, there's a number of targets for which either ligands or affinity resins are already available, depending on the application. And there is, as said, the opportunity to enter in a collaboration to make further specific chromatography resins meeting your needs. Another example of applying this technology is the C-tag system. So it's actually a very, very small tag which only consists of a natural sequence of 4 amino acids, which have to be on the C terminus. So it's hardly a tag in as much as it's 4 amino acids which could have been there on the C terminus of any recombinant protein such as VLP, and it's currently in successful implementation in a clinical program for VLP-based vaccine for human use. So it's something that can also be an opportunity to much simplify the downstream processing of your own VLPs or other adherent cells-made biologics. The other technology which you can benefit from is known as the POROS bead, which is described here very, very briefly. It's a backbone for chromatography, for large-scale chromatography, which has an absolutely unique structure made of an extremely rigid plastic polymer. It's also highly proven in the industry. It's been around for 20 or 25 years and used in numerous commercial processes, and it has very desirable attributes for virus purification, in as much as there's absolutely immense throughputs that goes through the bead, which offer really superior interaction surface for virus particles, would that be in cation exchange mode, anion exchange, or even HIC, and in some affinity application. So as an example, the technology which is really revolutionizing AAV purification nowadays is POROS CaptureSelect AAV, because it's the CaptureSelect ligand which is immobilized on a porous bead, which combines the characteristics of both technologies, right? One of our latest launches in this area is high performance HIC chromatography, which comes with unprecedented salt tolerance and flow rates in particular. Obviously, it's extremely important to have a word about analytics, right? Would it be upstream or downstream? Obviously, all our knowledge and your capability to successfully make life-saving vaccines it comes down to the analytics you have, to the process analytical technologies available to keep processes under control and develop processes successfully. So stay tuned, because there will be specific webinars going more into the details of each of those analytical aspects. A key example can be the host cell residual DNA analysis. And in that area, there is a particularly successful kit which is known as ResDNA-seq, and offers extremely high sensitivity for residual host cell DNA, and very uniquely, it comes in a ready-to-use package with a genomic standard. So it really has established itself as the state-of-the-art in that key measurement. In the area of protein impurities, such as host cell proteins, you may want to hear about Protein-seq, which is a successful hybrid of ELISA and qPCR technologies. And what does it do better than ELISA? It allows you a far broader dynamic range. So it's extremely useful, in particular in purification process development, when a high number of samples will come to your laboratory for HCP testing. And obviously, they're all of unknown content, and ELISA has a narrow dynamic range. In order to avoid having to do massive amounts of dilutions and retesting and let this analytical aspect become a bottleneck, Protein-seq is the technology that allows far more samples to come into the lab, get tested right the first time at the incoming concentration straight from the PD lab, and streamlined downstream process development. All right, so thanks very much for your attention on that last overview related to downstream purification and analytics capabilities. A very important point before we move to the Q&A is to let you know that we will very shortly be closing registrations for our workshop next week. So if this quick browsing of capabilities have been of interest for you and you want to hear more on the same theme, next week in Cramlington, UK, so close to Newcastle upon Tyne, we will have a full day of technical workshop with an industry speaker who has been employing those technologies in their Irish company. And we're going to have senior product management available as well, and the opportunity to also see many of the equipments used for those adherent cell workflow, as well as visit single-use technologies manufacturing plant there. The final registrations are going to close very, very shortly. So if you are interested and available to join us in Newcastle, UK next week on the Thursday, I really strongly encourage you to take your shot right now at trying to grab one of the remaining seats and benefit from that very interactive workshop, right? So with that, we may want to move on to some questions in the last minutes. Okay, so I see we have a question from a participant called Nila Vranjan. So with about sharing the documents. So yeah, Nilab, if you do provide us with a professional email address, I see that it's a Gmail address you've been using so far. So that would not be very compliant. But if you are willing to share with us any professional email address, we'll be more than happy to provide you with the content that you require. And please do not forget to find on the webinar interface, you should have access to some resources and be able to download an important part of the content. All right. So maybe in the last minute, maybe I would have one quick final comment to make, to invite all the participants of today's webinar that there will be an on-demand version of the webinar, and that will be absolutely possible to download. Stay tuned about that, right? So that's the last minute of the webinar. I just want to maybe give a quick word about the poll answers. I don't know, Céline, if you want to comment on the poll results briefly.
Yes, sure. So what is your main challenge regarding serum use in cell culture? Most of you have answered performance and log pre-qualification, and then availability and price, and finally logistic, storage and control. These are the three main factors that we have also identified. Surprisingly, viability is not a key point for the audience. For the second question, what is the alternative to serum to address this issue? Most people would rather change production cell line, or maybe wouldn't change because there's none of the options offered. And finally, why is adherent cell culture your process type of choice? 50% of you answer that it's necessary for the virus you're producing, which is very common. And secondly, the high cost of transitioning to suspension is hindering some of you in your change from adherent to suspension.
All right. Thanks very much, Céline, for those comments, and thanks very much to all poll participants. So it is time. It's important that we respect the timing that you have allocated. So we really want to thank all participants for taking part in that webinar. Stay tuned because we will be launching more in the near future. And of course, you can reach out to your Thermo Fisher Scientific representative for any further discussion, or to the speakers of the webinar by leaving your details. So thanks very much, and we hope that you appreciated this webinar and that it will help you successfully manufacture life-saving or otherwise important biologics from adherent cells in the future. Thanks very much. Have a great rest of the day, and talk very soon.